Chapter 17: Cancer and its interactions with the immune system. Cancer results from mutations that cause uncontrolled cell growth. This figure shows the difference between a benign tumor and a malignant tumor. In an adenoma, neoplastic glandular cells grow abnormally but remain confined within a fibrous connective tissue capsule. The surrounding normal tissue structure is maintained. In contrast, in an adenocarcinoma, cancerous glandular cells invade surrounding tissues, breaking through normal gland structures and spreading uncontrolled. This transition from localized, non-invasive growth to invasive behavior marks the progression from benign to malignant cancer. This figure highlights the common sites where cancers develop in males and females. In males, cancers frequently arise in the lymph nodes as lymphoma, in the lungs, pancreas, bladder, prostate, rectum, and bone marrow as leukemia. In females, common sites of cancer include the skin as melanoma, the breasts, kidneys, colon, ovaries, and uterus. These patterns reflect how different tissues and organs are vulnerable to the effects of mutations that cause uncontrolled cell growth. A cancer arises from a single cell that has accumulated multiple mutations. Cancer develops through a stepwise process involving the accumulation of multiple mutations. The sequence begins with a normal epithelial cell that loses a tumor suppressor gene such as APC, leading to excessive epithelial proliferation. Activation of an oncogene like RAS promotes the formation of a small benign tumor. Further loss of tumor suppressor genes such as DCC and P53 drives the growth of a large tumor and the transition to an invasive state. As the cancer progresses, rapid accumulation of additional mutations enables metastasis, allowing cancer cells to spread to distant tissues. Exposure to chemicals, radiation, and viruses facilitates progression to cancer. Certain viruses are strongly associated with the development of human cancers. Among DNA viruses, papillomaviruses are linked to genital warts and carcinoma of the uterine cervix, with a worldwide distribution. Hepatitis B virus is associated with hepatocellular carcinoma, most commonly in Southeast Asia and tropical Africa. Epstein-Barr virus is linked to Burkitt's lymphoma, nasopharyngeal carcinoma, and other B-cell lymphoproliferative diseases, particularly in West Africa, Papua New Guinea, southern China, and immunodeficient populations. RNA viruses also contribute to cancer development. Human T-cell leukemia virus type 1 is linked to adult T-cell leukemia and lymphoma, especially in Japan and the West Indies. Human immunodeficiency virus and human herpes virus 8 are associated with Kaposi's sarcoma, particularly in Central Africa. Certain common features distinguish cancer cells from normal cells. Cancer cells possess a distinct set of characteristics that differentiate them from normal cells. They stimulate their own growth, ignore signals that would normally inhibit cell proliferation, and avoid programmed cell death through resistance to apoptosis. Cancer cells also develop their own blood supply by promoting angiogenesis, allowing for sustained growth. They acquire the ability to invade surrounding tissues and spread to distant sites through metastasis. Furthermore, they replicate continuously and evade detection or destruction by the immune system. Immune responses to cancer have similarities with those to virus-infected cells. This graph illustrates the relationship between tumor cell population growth and tumor size over time. As breast cancer cells undergo successive doublings, the tumor grows exponentially. A tumor becomes first detectable on X-ray imaging when it reaches approximately 100 million cells. It becomes palpable as a mass at around 1 billion cells. If left untreated, continued growth leads to a lethal tumor burden at around 100 trillion cells, resulting in the death of the patient. This figure highlights how cancer can grow silently for many doublings before detection. Allogeneic differences in MHC class I molecules enable cytotoxic T cells to eliminate tumor cells. This experiment demonstrates the role of MHC class I molecules in tumor rejection. Tumor cells isolated from a primary tumor in one mouse strain are injected into two different groups. When tumor cells are transplanted into genetically identical mice with the same MHC molecules, the tumors grow and the animals eventually die. However, when tumor cells are injected into mice with different MHC molecules, the immune system recognizes the cells as foreign and eliminates them, allowing the animals to survive. This highlights the importance of cytotoxic T cells and MHC class I recognition in controlling tumor growth. This image shows a Tasmanian devil affected by transmissible facial tumor disease. The tumors are spread between individuals through direct contact, such as biting. Because the tumor cells share a very similar MHC profile with the host, the immune system fails to recognize the cells as foreign. As a result, the cancerous cells evade immune detection and grow unchecked, leading to severe facial deformities and death. This disease illustrates the critical role of MHC differences in enabling cytotoxic T cells to eliminate tumor cells. Mutations acquired by somatic cells during oncogenesis can give rise to tumor-specific antigens. When a normal cell undergoes mutations and becomes cancerous, changes in its proteins can lead to the production of new antigens. Some tumor cells present mutant peptides derived from altered cellular proteins, creating tumor-specific antigens. Other tumor cells reactivate embryonic genes that are normally silent in differentiated adult cells, leading to the expression of tumor-associated antigens. Tumors may also overexpress self-proteins, increasing their presentation on MHC class I molecules and enhancing recognition by cytotoxic T cells. These mechanisms allow the immune system to detect and potentially eliminate tumor cells. This table lists examples of mutated tumor antigens associated with different cancers. In melanoma, mutated forms of MERT2 and triosulfophosphate isomerase present altered peptide antigens. Non-small cell lung carcinoma and head and neck squamous cell carcinoma are associated with mutations in the ME1 and P53 proteins, respectively. Bladder tumors present mutations in KIAA205. In chronic myelogenous leukemia, the BCR-ABL fusion protein creates a novel peptide recognized by the immune system. Each mutated antigen is presented by specific HLA molecules, which determines how tumor cells are detected by cytotoxic T cells. This figure shows how a novel tumor-specific antigen can be generated through peptide splicing. In melanoma cells, two non-contiguous segments of the GP100 protein are cut out and spliced together by the proteasome, creating a new peptide that does not exist naturally in normal cells. This spliced peptide is then presented on the surface of melanoma cells by HLA-32 molecules. Patient cytotoxic T cells recognize this spliced peptide as foreign, enabling an immune response directed specifically against the tumor. Cancer/testis antigens are a prominent type of tumor-associated antigen. Cancer/testis antigens are a prominent category of tumor-associated antigens. These proteins are normally expressed only in the testis, but become aberrantly expressed in various cancers. Examples include MAGA and MAGIP, located on the X chromosome, and BAGE, located on chromosome 13. Other cancer/testis antigens, such as GAGE1, SSX, NYESO1, and LAGE are also found on the X chromosome. Because they are usually hidden from immune surveillance in normal tissues but appear on tumor cells, cancer/testis antigens serve as important targets for immunotherapy. Successful tumors evade and manipulate the immune response. Initially, tumor cells express high levels of MYC proteins on their surface. These proteins are recognized by NKG2D receptors on natural killer cells and gamma delta T cells, leading to immune-mediated killing of the tumor cells. However, variant tumor cells can evade this detection by cleaving MYC from their surface. The released soluble MYC binds to NKG2D receptors on lymphocytes, downregulating their activity. As a result, the variant tumor cells escape immune surveillance, survive, and continue to proliferate, ultimately leading to cancer progression. This image shows a tumor tissue sample stained for immune markers. Tumor cells are densely packed and exhibit variable expression of immune-modulating molecules. Areas stained brown indicate regions where the tumor has adapted to manipulate the immune environment, likely suppressing effective immune attack. Successful tumors often escape immune control by altering the local immune response, allowing continued growth and progression. This figure illustrates how tumors suppress the immune response to avoid elimination. Tumor cells secrete immunosuppressive cytokines such as TGF-beta, which inhibits the activity of cytotoxic T lymphocytes and T helper 1 cells. At the same time, TGF-beta promotes the activation and expansion of regulatory T cells, which further secrete TGF-beta and interleukin-10 to dampen immune responses. This tumor-induced immune suppression allows cancer cells to evade detection, survive, and continue proliferating despite the presence of an active immune system. Vaccination against human papillomaviruses can prevent cervical and other genital cancers. This table summarizes the major proteins encoded by human papillomaviruses and their functions. L1 and L2 form the viral capsid, with L1 as the major structural protein and L2 as the minor one. E1 functions as a DNA helicase, and E2 acts as a transcription factor that regulates viral gene expression. E4 is a structural protein produced in infected keratinocytes. E5 serves as a cofactor for epidermal growth factor signaling and regulates MHC class I expression. E6 promotes degradation of the tumor suppressor protein p53 and activates telomerase, while E7 binds to the RB protein, leading to the release of E2F and promoting uncontrolled cell division. These viral proteins contribute to the development of cervical and other genital cancers, highlighting the importance of vaccination to prevent infection. Human papillomavirus vaccines are available in two major forms, a bivalent vaccine and a tetravalent vaccine. The bivalent vaccine protects against human papillomavirus types 16 and 18, which are responsible for the majority of cervical cancers. The tetravalent vaccine protects against types 16 and 18, as well as types 6 and 11, which cause genital warts. The bivalent vaccine uses aluminum hydroxide as an adjuvant, while the tetravalent vaccine uses amorphous hydroxysulfate. Both vaccines provide about 70% protection against cervical cancers, and the tetravalent vaccine also offers 90% protection against genital warts. Vaccination with tumor antigens can cause cancer to regress, but it is unpredictable. This graph shows the response to cancer vaccination using a tumor-specific antigen. Early after vaccination, the patient's tumor progresses despite rising levels of antigen-specific cytotoxic T cells. Over time, an increase in T cell numbers correlates with tumor regression. Eventually, the patient shows no evidence of cancer. However, the response to vaccination is unpredictable as different individuals vary widely in how their immune systems respond to tumor antigens and control tumor growth. Monoclonal antibodies that interfere with negative regulators of the immune response can be used to treat cancer. This figure shows how monoclonal antibodies targeting immune checkpoints can enhance cancer treatment. Normally, T-cell activation occurs when the T-cell receptor and the CD28 co-stimulatory receptor both receive signals from antigen-presenting cells. Over time, the B7 molecule preferentially binds to CTLA4 instead of CD28, which inhibits T-cell activity. Treatment with an anti-CTLA4 antibody blocks this inhibitory interaction, allowing B7 to continue engaging CD28 and maintaining persistent T-cell activation. This strategy boosts the immune system's ability to attack and destroy tumor cells. T-cell responses to tumor cells can be improved with chimeric antigen receptors. This figure shows the structure of a chimeric antigen receptor used to improve T-cell responses against tumor cells. The chimeric receptor combines an antigen-binding domain derived from antibody variable regions with intracellular signaling components from CD28, CD137, and the zeta chain. This design allows T cells to directly recognize tumor antigens without the need for antigen processing and presentation by MHC molecules. Upon antigen binding, the receptor delivers strong activation signals, enhancing T cell proliferation, survival, and cytotoxic activity against tumors. This figure illustrates adoptive cell transfer therapy using effector T cells engineered to express a chimeric antigen receptor. The engineered T cells express receptors that recognize CD19, a surface protein found on B cell tumors. Upon binding to CD19, the chimeric antigen receptor delivers strong activation signals through CD28 and the zeta chain, independent of MHC class II interactions. This direct recognition enables rapid activation of the T cells and targeted killing of tumor cells, making chimeric antigen receptor T cell therapy a powerful strategy for treating certain cancers, particularly B cell malignancies. The anti-tumor response of T cells and NK cells can be augmented. This figure shows how natural killer cells can eliminate tumor cells through antibody-dependent cellular cytotoxicity. Therapeutic monoclonal antibodies bind to specific tumor cell antigens. The Fc portion of these antibodies is then recognized by Fc gamma receptor 3 on the surface of natural killer cells. Once engaged, the natural killer cell releases cytotoxic granules that directly kill the tumor cell. This strategy enhances the anti-tumor response and is a key mechanism behind the success of several antibody-based cancer therapies. T-cell responses to tumors can be improved by adoptive transfer of antigen-activated dendritic cells. This figure describes how T-cell responses to tumors can be enhanced by adoptive transfer of antigen-activated dendritic cells. Monocytes isolated from a prostate cancer patient are cultured with a fusion protein composed of granulocyte macrophage, colony-stimulating factor, and prostatic acid phosphatase. This fusion protein drives the differentiation of monocytes into dendritic cells, which process and present prostatic acid phosphatase-derived peptides on MHC class II molecules. The activated dendritic cells are then infused back into the patient, where they migrate to lymphoid tissues, such as the spleen, and stimulate naive T cells, boosting the patient's anti-tumor immune response. Monoclonal antibodies are valuable tools for the diagnosis of cancer. This figure shows how monoclonal antibodies and molecular techniques are used in cancer diagnosis. Hematoxylin and eosin staining reveals the general cellular architecture of a tumor. DNA hybridization with an anti-MYC oncogene probe detects genetic abnormalities associated with MYC amplification. Staining with an anti-MYC oncoprotein antibody identifies cells overexpressing the MYC protein, helping confirm oncogene activation at the protein level. Together, these tools provide precise diagnostic information to guide cancer classification and treatment decisions. This figure highlights how different monoclonal antibodies assist in the diagnosis of cancer. Hematoxylin and eosin staining provides basic visualization of cellular morphology. Immunostaining with anti-PAX5 antibody identifies B-cell lineage tumors. Anti-CD30 antibody staining is used to detect Hodgkin and Reed-Sternberg cells in Hodgkin lymphoma. Anti-CD15 antibody staining further characterizes certain lymphomas by highlighting additional tumor markers. These targeted stains allow for more precise classification of cancers, improving diagnostic accuracy and guiding treatment selection. This figure demonstrates how monoclonal antibodies are used in cancer diagnosis. Hematoxylin and eosin staining provides an overview of tumor cell structure. Immunostaining with anti-CD30 antibody highlights the presence of CD30-positive tumor cells, common in Hodgkin lymphoma and certain T-cell lymphomas. Staining with anti-NPM-ALK fusion protein antibody identifies tumors with the NPM-ALK translocation, a genetic alteration seen in anaplastic large cell lymphoma. These targeted stains improve diagnostic precision and inform appropriate treatment strategies. Monoclonal antibodies against cell surface antigens are increasingly used in cancer therapy. This figure lists human monoclonal antibodies used in cancer therapy. Naked antibodies like trastuzumab target breast cancer by inhibiting ERBB2 signaling, while bevacizumab treats colon cancer by blocking vascular endothelial growth factor. Cetuximab and panitumumab inhibit epidermal growth factor receptor signaling in squamous cell carcinoma and colorectal carcinoma, respectively. Ipilimumab is used for melanoma by inhibiting CTLA-4 signaling. Rituximab, alemtuzumab, and ofatumumab target B-cell malignancies by promoting antibody-dependent cellular cytotoxicity or complement-dependent cytotoxicity. Conjugated antibodies such as brentuximab vedotin and 90Y labeled ibritumumab deliver cytotoxic drugs or radioactive isotopes directly to tumor cells, enhancing tumor cell destruction while limiting systemic toxicity. This figure explains how targeted delivery of a cytotoxic drug can kill lymphoma cells. An anti-CD30 antibody is chemically linked to auristatin, a cytotoxic drug, by a cathepsin cleavable linker. After the antibody binds to CD30 on lymphoma cells, the conjugate is internalized. Inside the cell, cathepsin enzymes cleave the linker, releasing auristatin. Auristatin disrupts microtubules inside the cell, preventing formation of the mitotic spindle. As a result, the lymphoma cell is unable to divide and eventually dies by apoptosis. This figure shows how a monoclonal antibody linked to a radionuclide can target and destroy cancer cells. An anti-CD20 antibody is conjugated to a radioactive isotope. After binding to CD20 on the surface of malignant B cells, the radioactive antibody delivers localized radiation that damages the cell's DNA. The radiation-induced DNA damage ultimately kills the tumor cells while minimizing harm to surrounding healthy tissue. This targeted therapy is used to treat B-cell malignancies such as non-Hodgkin lymphoma.